What are type of nucleotide databases? Let's start. There are several types of nucleotide databases available, each serving different purposes and containing different types of information. Some examples include genomic databases. These databases contain information about the genomic DNA sequences of organisms, such as GenBank and the European Nucleotide Archive (ENA). These databases are used to study the genomic architecture and evolution of organisms, and to identify genes and other functional elements in genomes. Transcriptomic databases. These databases contain information about the mRNA sequences of organisms, such as the RNA-seq data from the Genomic Data Commons (GDC) and the Cancer Genome Atlas (TCGA). These databases are used to study gene expression patterns and to identify alternative splicing and post-transcriptional modifications of RNA. Variation databases. These databases contain information about genetic variations such as single nucleotide polymorphisms (SNPs) and small insertions or deletions (indels), such as the DBSNP and the 1000 Genomes Project. These databases are used to study the genetic diversity of populations and to identify genetic factors associated with diseases. Epigenetic databases. These databases contain information about the modifications of DNA and chromatin that can affect gene expression without changing the DNA sequence, such as the Roadmap Epigenomics Project and the Encyclopedia of DNA Elements (ENCODE). These databases are used to study the regulation of gene expression and the role of epigenetics in development and disease. Regulatory element databases. These databases contain information about non-coding regions of the genome that are involved in regulating gene expression, such as the Transcription Factor Database (TFDB) and the JASPR database. These databases are used to study the mechanisms of gene regulation and to identify cis and trans-acting elements in genomes. Functional annotation databases. These databases contain information about the functional elements in genomes, such as genes, exons, introns, promoters, enhancers, and regulatory regions. These databases are used to annotate genomes and to study the functional organization of genomes.